Hello, everyone. Hello. Welcome to our 9th DHEP, Deaf Hub Exploring Perspectives in Biomedical Science lecture series. Today we will have Victoria Popov, or Tori, presenting for us. Tori is an alumna of RIT. She received her bachelor's degree in Biomedical Science in 2017, and then she continued in the Bridges to the Doctorate program and completed her master's in professional studies in 2019. She's now a fifth-year PhD student in neuroscience at the University of Rochester, working with Dr. John Fox and Dr. Ed Freeman. She was recently awarded a very prestigious grant from the NIH, the F99 K00 award, which will fund her as she completes her PhD and continues her research as a postdoc. Now let's all give a warm welcome to Tori. Welcome, Tori.
It's very exciting to be here today. I hope I'm speaking loud enough. Can people hear me in the back of the room okay? All right. I hope my voice doesn't crack, so I have my water here, but we'll go with the flow. So as Dylan mentioned, I'm currently a PhD student in the neuroscience program over at the University of Rochester. So today I was really thinking a lot about how, the things I wish I knew when I was an undergraduate student. So I really hope that what I share today will be beneficial. But of course I am biased. I love the research that I do. So I'm also going to talk a little bit about that because I think it's really important to see different ways how you can talk about research and ultimately see opportunities for yourself going forward if that's something of interest to you. So I just wanted to ask real quick, I know that the RIT now has a new neuroscience program for the undergrads. There's also a cognitive science PhD program, which is absolutely phenomenal. Are any of you guys in those programs? Yeah? Neuroscience, that's awesome. Well, I'm glad that you guys could make it. Please feel free to ask any questions you have throughout the talk. It doesn't have to be a wait to the end. Just jump right on in because I want this to be discussion focused. So just to talk a bit about my journey, you can see a couple of pictures here. So when I was a young girl, I loved horses. It started with a gift that my aunt gave my brother and I. We had a month of horseback riding lessons that we got for Christmas. And that really stuck with me. I still horseback ride to this day. And I spent a lot of time at the barn. That was something I was really passionate about. I was even a working student, and I would help whenever the veterinarian would come with any procedures that she would perform with the horses. And this was really the start of when, I always thought my career path would be to become an equine veterinarian. We know how life goes, and that's not always the case. But I also included a picture of myself with my childhood dog. She's half German Shepherd, half Lab. And the reason for this is because I think back to late elementary school when I had my first real science experiment. So we had to come up and apply what a hypothesis is, have a research question, and then have your results and finding. So I wanted to see, I guess she could catch the ball 100% of the time. That wasn't quite the case. It was more like 85%, if I recall correctly. And that was, the first science experiment. So the reason why I share this is because I remember for as long as I could remember that science was something, science being a broad term, that I was really passionate about. And I thought that this would be in the case of working with horses. So fast forward, I came to RIT, and I was from 2013 to 2017. As Dylan mentioned, I got my bachelor's in biomedical sciences, and I was on the pre-med track. But I do want to step back. This is actually when things started to change a little bit. So I had a really hard time. I learned detaching my emotions from the animals from a medical perspective. So I knew that veterinary medicine wasn't for me. But I still had this passion for science, and I wanted to figure out what does that really mean to me. So I did a bunch of different things when I was at RIT. This picture here is from the human gross anatomy class that I took. Is anyone taking that class? Do they still offer it? They do. It's a great class. I highly recommend taking it if you can. Even if you're not interested in the medical school route, it'll be very beneficial for a neuroscience PhD program. And this is the first time, if I look back, at the time I didn't know, but looking back, that would be the first time I really started to open my eyes and become interested in the brain. And I wanted to also add these couple pictures at the bottom because I think support is something that people talk a lot about when you're navigating life. And for me, I'm thankful that I have a very good support system from my family. So this is just a really cute picture of a book that my dad gave me when I graduated in 2017, just saying to enjoy the journey. So it's something that really stuck with me, and I hope that you guys remember that even when you're in the trenches. So while I was at RIT, I was working on the pre-med route. I did go as far as applying to medical schools. I did primary applications. I'm not sure if people are familiar with the medical school application process, but then I realized I loved being in the OR. I had a lot of shadowing experiences in different fields, from orthopedics, pediatrician, cardiology, and I realized I loved the time I spent in the OR, but if I'm being completely honest with myself, I had a hard time getting past certain situations where I would feel squeamish. So I had to really step back again. I knew I loved science as a broad term, but for a second time I had to ask myself, what does this really mean to me? So I thought I was going to go down the ENT route with medical school, and then I wanted to further explore my research opportunities, and that's where the opportunity to join Bridges and focus on a more research perspective with science came into play. And I should add that after my third year, during my time as a student at RIT, I went to the University of Michigan. So I was in the Cressy Hearing Research Institute, and I did some otology research when I was over there. Highly recommend, if you can, get as much research experience, even if you don't think it's for you, during your time at RIT, because that's the time to explore if you're interested in science and research, all these different tiers, health medicine, it's all going to tie together somehow, and it's your story to tell. So fast forward, like I mentioned, I joined the Bridges program, and during the first year of my time at the Bridges program, some of you may know, my research was focused on research at the RIT. Then the summer between the second year, I did research at the University of Rochester. So I really wanted to see if ear research was something that interested me. If I'm being completely honest, I noticed my passion just wasn't there. I was working with mice. I was in Dr. Leah Mitchell's lab. Her research is phenomenal, but I just learned quickly that that wasn't something that was for me, and that's okay. I realized I wanted to get that connection with humans again, so I wanted to go more into human participant-focused research. And that's where I got to do research with Peter Hauser, Jessica Gulick, some of the awesome people in their center. So we worked with individuals who were deaf or hard of hearing, really trying to understand language abilities, health disparities. And it's great now because NIH actually this year finally recognizes that there are health disparities in the deaf and hard of hearing population. So it's nice to see these changes being rolled forward, even though we knew that so many decades ago. So I did the Bridges program. Then the second year, I did research over at the University of Rochester. I continued to work with human participants, but from a more clinical perspective. I should highlight that my main thesis project as a master's student when at RIT was in the Department of Psychology. So I worked with human participants who had schizotypy, which means that they were at a higher risk for developing schizophrenia later in life. So that was my main thesis project, and I really had a passion I found for this population, so I continued this research when I was over at the University of Rochester. And that's where I started to develop more neuroimaging skills. So I had exposure to MRI, working with this clinical population of people with schizophrenia. And that's where the passion really started. And this was something that I wanted to see to the end. When I finished with the Bridges program, I did apply to PhD programs in clinical psychology. And I did not get accepted the first time, but that is completely okay. I know they say in the moment of, it's really hard to see what the future holds, but you just have to keep putting yourself out there. So I had to figure out, what's my next step? I'm gonna reapply in a year, but what's, the plan for that coming year in between? So I went ahead and I reached out to people that I knew to see if there were potential openings in labs, job positions, you name it. And the other half of the time, I was actually doing more of a deep dive online to find these job positions. And I'll never forget, there was one day I almost did not send this email, and that was the email that changed everything. So I emailed someone who was in the lab that I am currently in now for my PhD thesis, and that person got me in touch with one of their PIs who runs the lab. And then that person, I did have to send a follow-up email, send those, keep bugging these people at the top, they're very busy. And they told me about the University of Rochester PrEP program. So it's a program not just at the University of Rochester, but all across the United States. There are different clinics and universities that have this program. It's NIH funded, and the goal is to get underrepresented minorities into PhD programs. So it's a similar concept to Bridges, just focusing on not only those who are deaf or hard of hearing. And through this program, in the laboratory I mentioned of the person that I emailed, that is when I designed my now thesis study that I'm going to talk about. And I had to get it designed, really critically think, IRB approved, and then fast forward, we have all this data we can go through. So I just wanted to highlight this, and I really like the picture on the bottom right, just because it's October. So for those of you who know what EEG is, it allows us to measure brain activity across the scalp. So of course, we didn't see much activity through the pumpkin, but it was just a cute way to celebrate the festives of the month. So in a very simple lay term, I'm going to talk a little bit about my thesis, and then I'm going to move on to things I wish I knew or did differently, as well as opportunities that I could think of and put together for you guys in research. So the big point of my study is we want to understand, sometimes you go for a walk and your brain clears up, so you feel better after the walk. The big idea is what's going on in the brain while you're walking that is clearing up that brain fog. And brain fog can be a very big term, but I usually focus on the term response inhibition. That's one of the many different types of executive function. And I'll explain momentarily on the next slide why I'm interested in that component. So we're hoping to see if walking could be a potential therapeutic supplementary for people with schizophrenia. So we know that we are constantly engaging with our everyday environment, and that's something to be very considerate of. In people with schizophrenia, they have a hard time being able to inhibit certain behaviors. So I think of one example from a patient that I work with who shared this was something she didn't recognize. It was feedback she got from her sister. But a month before she heard her first voices and saw her first visual hallucinations, she reported that she was spending a significant more amount of money that she didn't have making Amazon orders. So she had a ton of packages coming up to her place. So this is a good classic example, I think, of not being able to have that inhibition, and that's an early target, and that's why I'm particularly interested in this response inhibition component, because it's an early intervention therapeutic target for this population. And as far as treatment goes, I do want to highlight that individuals with schizophrenia have many different characteristics, which I'll talk about on the next slide. But as far as we know, current treatment for individuals with schizophrenia is cognitive behavioral therapy coupled with antipsychotic medication. But the issue is this therapy is really only helping with the positive symptoms, those added on sensory experiences, but it's not helping with the negative or the cognitive symptoms. So there's a big push in the field to really understand if we can come up with supplementary treatment to help the cognitive and the negative symptoms of people with schizophrenia. There are many different subtypes of schizophrenia. I'm not going to go into all of these, but I just wanted to highlight that I'm going to be talking about schizophrenia for today's talk, but there's also other subtypes, and what's common between these subtypes is that impulsivity deficiency. Does anyone know what schizophrenia is? Out of curiosity, have you studied about it in your class? I'm sure you have. I just want to make sure that we're all on the same page, so that's why I want to include this slide. So we know that of the different characteristics, individuals diagnosed with schizophrenia can experience delusions, which is the unshakable theory or belief in something that is not true. You can also have hallucinations, the added on sensory experiences, like seeing or hearing things that aren't there, as well as disorganized speech. So sometimes patients will report that they may have someone tells them that they're speaking sort of like in a Yoda format, the context of a sentence just isn't flowing as correct. And you can also experience catatonic behavior. So I like to think of this as muscle rigidity. And this can be due to schizophrenia, but it can also be due to the medication itself, as well as the negative symptoms. So that's a removal of emotion or experience you can have. So sometimes, I'm not sure if we have many people who are gamers in the audience, but usually when you're playing these games, you're enjoying them. So with schizophrenia, you may not have that pleasure or enjoyment during an activity that you really like. So to be diagnosed with schizophrenia, you do not have to have all of these symptoms. These are just categories of certain symptoms people may experience. You really just need two, and one of them has to be either delusions, hallucinations, or disorganized speech. So as far as the literature goes, why am I doing my study? We know that there have been findings that people with schizophrenia have a response inhibitory control deficit. We also know that before and after an exercise intervention, that there's improved executive function. And the third piece is there have been studies that have looked at brain activity in people without schizophrenia and have seen improvement in inhibitory control during an exercise intervention. But no one has looked at brain activity in people with schizophrenia during the exercise intervention to see why there's this improvement in inhibitory control. So that's where my study comes in. What's going on in the brain that's leading to this improved inhibitory control, if at all. So we are changing science in some way. I'm not sure if anyone, you can do a show of hands, has anyone worked with EEG? Yeah? You had someone in the audience? Okay. Well, for those who don't know, when doing neuroimaging studies, this includes MRI, EEG, usually you want people to sit still, and this is because you don't want to add noise to your data. You really want to make sure you're looking at brain activity. But we're actually saying, we're going to change science. We want you to move, we want you to walk on our treadmill. This is more naturalistic. As you know, as humans, we're constantly engaging with our everyday life and our environment. So the way we are able to do this is through our mobile brain body imaging, or MOBI, apparatus. Very fancy term. It really just means that people are walking on a treadmill, wearing an EEG cap, a jumpsuit for kinematics, and completing a task on the screen. So we get 3 data streams, which I'll talk briefly a little bit about today. If you guys have questions at any time, you of course feel free to check in. I want to make sure I'm going forward right. And just to really clarify here, when we're looking at brain activity, this is, you can see the blue line is the average. So all the red lines that are appearing are just each individual participant. So we're curious about group averages. So for example, those without schizophrenia and those with schizophrenia. So the big point here is there's 3 components we know from these electrical activity that's recorded across the scalp with our EEG that tell us different meanings. The first part is related to visual processing. So this is likely when, for example, while doing the task on the screen, you see a picture. So about 100 milliseconds after, that's when your brain would have processed that it saw the picture. Then there's 2 different parts that are related to response inhibition. The first part is the automatic component. Second part, cognitive component. That sounds complicated, I know. I like to think of it in the sense, very simply put, that it's the idea of your brain knows when they should or shouldn't press a button, and then you would know. So it takes time before your brain has to process that. So what did people do while they were in our apparatus? So we had them do a response inhibition task. So most of the time, participants were pressing the button. Then every once in a while, we say, don't press the button. So if a picture is different, press, same, don't press. And it's those times when participants don't press that I was really interested in, because that's the inhibition. You're stopping yourself. And as you can imagine, there are many outcome variables, the times you're correct, the times you weren't correct. And I do want to highlight that throughout the experiment, participants were not always walking. Sometimes they're sitting. So we had random motion states going back and forth between sitting and walking. And during the times we were sitting, trust me, I made sure I turned that treadmill off before I put the chair on the treadmill for those blocks. So there's two different ways. Just to circle back to the question, you can imagine while you're walking, doing this task, there's a lot going on as a participant from their perspective. And on the x-axis, I want to highlight the cognitive motor task load. That really just means doing the task while walking down the treadmill. And on the y-axis, that's related to how well someone was performing on their response inhibition task. Were they able to stop themselves from pressing the button when they were supposed to? So I'm curious what you guys think. And there's no wrong answer. Do you think that people will have a better time with inhibitory control doing the task when they're walking, or do you think it's going to be too much? Yeah. Who thinks they'll do better? You can raise your hand. Yeah. Who thinks they're going to do worse? Worse? Both happen, actually. So let's talk about it. I err on the side, I think that participants are going to do better. That's my hypothesis. So how well did people do on the task? I'm going to break this down a little bit. On the left, we have our controls. On the right, we have people with schizophrenia. And you can see that each line is one person comparing the difference between sitting and walking. So I hypothesize that when walking, people will do better. This fancy formula in the y-axis really just relates to inhibitory control performance on the task. The blue line are participants who did significantly better with the task when they were walking compared to sitting. Gray line are those participants who had no significant change. And yellow are those people who actually did worse when walking than sitting. So it's this idea in the field of cognitive integration versus interference. So now this is looking at the individual level, but we did find that when comparing between groups, the next slide has a statistical analysis test I did, but on average as groups, people with schizophrenia actually did worse on the task than those with healthy controls. So as far as actually doing the task, it doesn't support my hypothesis. We thought that when walking, people would have a better time stopping themselves from pressing the button. So we didn't see that from a behavior point. That's okay. Brain data is where things start to get exciting. So here we are looking at 3 different electrode sites. One's frontal, one's central, one's a little more on the occipital lobe in the posterior region. And the reason why I chose these 3 electrodes is because they're right over that response inhibition circuit. So the idea is we have our sitting and our walking conditions. So there's 2 lines for patients, 2 lines for controls. The blue and the purple is what you'd want to compare for the sitting. And what I should add is this is for all the times participants were correct. And you can see with the gray shaded area, I talked about the visual processing, then the 2 components for inhibition. This take-home message for this slide is that we were able to replicate that our patients with schizophrenia did have that inhibitory control deficit when they were sitting compared to the controls, yes. Now, what about when walking? So these were all the times all participants successfully stopped themselves from pressing the button. So what you can see is at 0, this is when participants would have seen a picture, the same one the second time, and they know, okay, this is the same. I'm not supposed to press the button. But what's going on in the brain? How did that look? So again, the patients for sitting and walking are purple and orange. Controls, blue and green. I don't expect you to memorize that in any way. But the big take-home message you can see here is that around the 0.3, that middle gray bar area, this was related to the automatic component of inhibition. The last gray bar was the cognitive component of inhibition. So what we're seeing is that the last bar, the neuroactivity, the amplitude, is starting to look like that of controls, but not so much with the middle gray bar. So this likely means that our patients, as far as understanding the inhibitory component, the neural activity is starting to look like that of controls when walking. So it's very promising. And keep in mind that they weren't walking consistently. Participants were going back and forth between sitting and walking. It was only 4 minutes each time. So the fact that we're starting to see these differences gives us that green light for a clinical trial. Figure out, if I had schizophrenia, I would want to know, how long do I need to walk for? How often? Is it 15 minutes, 30 minutes, 2 times a week, 4 times a week? So that's sort of the idea, is that we got this green light. The next step will be to iron out all those details. And just in case, to really drive home this message, this was a statistical test. So the blue and the red that you see in the center is related to the t-test. If it was yellow, that means that the peak amplitude when walking was greater than sitting. I should back up and highlight the right are patients with schizophrenia, left are those with controls. And this was looking across all electrode sites on the scalp. And we grouped them together. So if you can see on the y-axis at the top, it is actually red. And that's the frontal region of the scalp. And it goes all the way to more posterior. So this is looking at the whole scalp. And when there is a significant difference, that's when you saw the yellow versus blue. But the big point is you can see that neuroactivity in people with schizophrenia is starting to look like those of controls. It's just happening later. You don't see much in the visual processing region. So that's the big take-home message. Last data stream, just to talk about it before we dive into more engaging discussion, was the kinematics. So this is really fascinating because there's very few studies out there that have been able to capture the movement of people with schizophrenia. I mentioned one of the characteristics that this population can experience is Parkinsonism. So that abnormal movement of muscle rigidity. So this is just for one control in one patient. And then the next slide has more all the participants. But I thought this was quite representative of what we saw in our groups. So you can see there's 3 axes here. They really just refer to, we looked at a left heel marker. It refers to the up and down, but also sideways movement of that marker on your left heel. So the big point is you can see with controls, they had a pretty smooth step characteristic. But when looking at patients, there's a little bit more of a sway. You could see as more, not as smooth. And this is representative here as well. Each line is a participant again. On the left, this is when participants were engaging in their experiment. But we also had one block where they were just walking. So they didn't see pictures, they weren't doing the task. The big take-home message is when you're looking at stride distance, which is the distance between where the foot left the ground and then touched the ground again, our healthy controls had a much greater stride distance. So it was actually much shorter for our patients. So as far as my knowledge goes, this is the first time we were able to capture this motion information in people with schizophrenia. So it's very exciting. You can imagine there's tons of different questions we can answer. Just to summarize this, okay, so as far as brain activity goes, we saw significant differences in regard to how well people did on the task. Not everyone did as well as we thought. We thought that our people with schizophrenia would actually do better. But I do want to just make note that there were a couple people with schizophrenia who did benefit when walking. So the other idea, I didn't talk too much about it today, is why did those people benefit and not other patients? So I predict we have a lot of different assessments we do. One of the things that we found is that these 2 participants had a mild symptom severity. So that's indicative of, we need to figure out if there's other details for why these people were benefiting and others were not. We didn't see the change like we thought, but that's okay. And we did capture the differences from the kinematics and the brain activity standpoint. I know I just spewed a lot of research data at you. This is a study that I'm very passionate about, and I think it's quite novel. not many people know of our Mobi apparatus. And I do want to highlight that there's actually 5 locations in the entire world who have this apparatus. There are studies that look at brain activity while people are doing an exercise, but what really makes Mobi special is that we're also getting kinematics, data, motion. And I always tell my participants when they come to the lab, it's almost like you're going to Hollywood, you get to be an action star, because that Velcro suit with those kinematic markers is actually what they use in action movies. So it could look like someone's jumping off a cliff, but really they're not. They're just changing the backdrop. So that's what makes this special. And there are many different people I can thank. I'm not going to go too much into it. And then there's a second part to today's talk. I do want to open this up to questions related to the research. One thing in the second part of today's talk I'm not going to talk too much about is the grant writing process for the grant I was recently awarded. But I did want to talk a little bit more about that. I applied to this grant. I remember about a year ago, I was really in the throes of it for the first application cycle, which was due in December. And when I highlight, this is with the NIH process. How many of you guys have heard of NIH? Yeah, most of you guys. So this is one of the organizations that you can go through to get grant funding, but it's not the only one. There's others like NSF, so forth. So what I say, it's not always the same process. But for NIH, they have quarterly application cycles. So I was applying for the December cycle, and I didn't get this grant the first time, but I did get fantastic feedback from my reviewers. You never know what you're gonna get. You have to, the way it works is, so you apply. I applied December 15th, and I got a summary statement back, but that wasn't until March. So that was a few months later. It takes time for your application to make its way through all the different steps to get to the reviewers, and then their feedback has to be compiled together before that even comes to me. And when that comes to me, I've said it before, but it's the summary statement. So I had 4 reviewers. Some of them liked it. Some of them had feedback that they wanted to see before they would consider getting this funded. And I had a month to be able to get all their feedback in and then resubmit it. But I do want to highlight, I actually had a conference that was all the way in Florence, Italy. And so I was trying to get this done. A month wasn't a long time. And I remember being at this conference, and I was trying to get as much as I could done. I would go in the morning, work in the hotel room after. And, it's a great time. You do what you do. But the big point of why I'm sharing this is that I was successful the second time that I reapplied. I have heard that most of the time, people will have to reapply for grants. So don't be discouraged if you're someone who in the future is writing a grant, you're applying for it. It's a lot of time for something that may not even work out. But when it does, it is the best feeling because you get to do the research that you want to do. So I'm sure maybe some of you have heard of the F31 grant. That is a grant that will fund your PhD research. And usually when you're in a PhD program, this is a grant you would apply for in your second or third year. I decided to go for the big one. So this grant is 6 years. It'll fund the last 2 years of my research during my PhD program, and then it'll fund 4 years of my research as a postdoc. What's exciting about it is this makes me competitive now because I will be looking for a postdoc lab because I would like to stay in the academia route, but now I have the funds to come with me. So when I join a lab, the lab will not have to pay for me. And I have become close with the privacy officer, who's a PO. This is someone within NIH that you work with a lot if you have questions related to your reviewer feedback, you name it. If you're applying for a grant, make sure you try to get to know your PO, just get your name out there, it can't hurt. And she has mentioned that they would love to see other deaf and hard of hearing people come through this program. So even if it's a couple years from now, don't be shy, reach out, I'm happy to talk to you. I would also love to see another person who's deaf or hard of hearing go through this program. So one thing that really drives me as I go into the postdoc position is I want to start to circle back to including deaf and hard of hearing people in my research. There's a ton of great neuroimaging studies that are going out there, but often, deaf and hard of hearing people are overlooked. So there have been a couple of studies that came out of a lab in the UK, and they reported lived experiences of people who were deaf and had a diagnosis for schizophrenia. And what we found is that these people viewed their hallucinations in their primary language. So if someone's primary language was ASL, rather than hearing voices, they would see someone signing to them as the voice, per se. So there's been no neuroimaging studies that have been done. So this is a gap that I'm really passionate about. And when I have my lab, in however many years, 6 years from now, it's going to be a long time, that's the first question I really want to answer. So I know that I talked about my study. I am biased in that sense because I think what I do is fascinating, but there's lots of different other studies that are going on that if you were interested in, there's different ways that you can get involved with it. So I know I mentioned the Bridges program. I'm happy to talk about that. And you can talk to other scholars who maybe are in the program now that you know of. There's also the PrEP program, which I mentioned. It's not just at the University of Rochester. That could be 1 year or 2 years. So if you were looking for a shorter research experience, because I know Bridges is 2 years, this could be another opportunity. And there's also the Summer Scholars Program. So I was very excited last summer. I actually had someone from RIT come join my study and get to know the whole pipeline. So that's another way to get a summer research experience. I know we were talking a little bit about that earlier. There's also these REU programs that you can apply for. These are just to name a few, but I just wanted to, if you found yourself passionate about research, just give back a little bit with pipelines that you can get involved with. This is a really text-heavy slide. I do not want you to read this or go into the nitty-gritty of it. You can focus on the 4 different columns. But I know that I talked a lot about my research. We're not just here to do research. We have other passions. We're human. We have work-life balance. So I wanted to touch on that. But in graduate school, I found myself really passionate about community outreach. I love the science I want to do, but I wanted to get children excited about it, to think about a future perhaps in neuroscience, especially if they have parents who weren't in science. I wanted to let them know this is an opportunity for them. So at the University of Rochester, another reason why I chose UR is because they have a Neuroscience Diversity Commission. And this was founded in 2020. And the big point is to get our science out there, give opportunities to people who may not have otherwise had it. So these 4 colors represent different work groups. The green is my work group that I'm involved with. That is Neuro to All. So we work with elementary to high school aged children. The next work group, Neuro East, works with high school students. So there are high school students in the Rochester area who actually come to a lab at the University of Rochester to do research. And then we have Neuro City, which is college students coming to do research. And we have Neuro Yes. So those are postdocs looking to find a job at a university. So one passion I really had was with Neuro to All. I joined the Diversity Commission when I joined the PhD program in 2020. And then because of my passion, I would show up to these meetings, really be a part of founding several things that came out of this work group. And now I am co-leader. So it's just something to consider. If you find yourself passionate about something, keep showing up and it'll likely come across that way. So this was that was the Neuro to All logo, but these are just different pictures. So what we do with Neuro to All is we have our pop-up events. We go out into the community and we have these scientist and training kits. So that's what really makes us special, because we have these hands-on activities that children get to go around and engage with, but they also get to do a brochure and these materials to do the experiment at home. So maybe their siblings, their parents can start to open their eyes to the science that we do. So this is just a bunch of different pictures, and I founded a class, and this class is how we get our new ideas, because we have students, whether they're undergrads, graduate students, that take the class and they learn all about We have guest lectures, how to talk about science in lay terms, how to engage with children, especially the rowdy ones. It's sometimes hard to get the attention of a child, and so forth. And they come up with their hands-on activity, they budget for it, we order the materials, then they piece it together, and we get to go out into the community to implement these hands-on activities. One example we had was, I'm trying to think of a good one here. So this is the Tasting the Rainbow. So it's teaching children about how the taste and smell olfactory system works. So we had children, they have a Skittle with their nose open, then try the Skittle again with your nose closed. So it's the idea that when your nose is closed, you're not able to guess the flavor of the Skittle. So it's the concept of you need both systems to work together to be able to taste. So very simple put. There were many other more complicated activities. I don't have pictures up here. We had one student who even used balloons, straws, cups, and water to show how the valves of a heart work. So that was pretty cool. Anything you are passionate about, you can definitely get it in lay terms, and you can do so through our course. One thing I want to highlight with this slide is I know that when people think of a PhD, they think of someone who's going to become a professor, teach at a university, have their own lab. And while that is the path I talked about and I am genuinely passionate about, that is not always the case. There are so many opportunities out there. One thing that has been of a hot topic discussion of late with me and my peers is how there seems to feel like there's a split between academia and industry. That split doesn't have to be there. If you have an idea, there are so many ways that you can bring that together. I know of several people, my PIs included, who have a lab. We do the research we do, but you can have contracts with pharmaceutical companies, these industry companies, with a common mission to, for example, get treatment for a certain population or just to understand what's going on at the neurophysiological level. So that's just one example, but I just want to highlight that not even all job opportunities with the PhD are on here. It's what you make of it. So if you're interested in becoming a PhD student, you really think about the skills you have, the skills you want to learn. You could do a skills gap analysis, and that'll help you figure out what you don't have and need to get to your next step. No one's perfect. You want to have those weaknesses. It shows that you're human, but you also want to have a plan to overcome that and constantly better yourself. So just wanted to highlight that. And I talked a lot about support network. I think we don't have to come up with an answer for this, but these are just two questions that I have constantly kept with myself if I felt that I was maybe not getting the support I needed, or I just felt like I was really in the trenches and I wanted to bring myself back up. These are two questions I felt were helpful for me to figure out if there's a gap and then go from there. So it could be, do you have two trusted people in your network? It doesn't have to be those in, more so in your career network. I think that's what's important. And then as well as, do you have that extracurricular? We're not all about, the research and the science, the studies that we do, but it's important to just get yourself out there, go rock climbing at Red Barn, go horseback riding, even just something as simple as just watching your favorite movie and take that time for yourself. We already talked about quite a bit of this in the discussion. I wanted this to be discussion-based, so that's why I'm able to skip over these slides. But I just wanted to, when networking, do as much as that as you can. You've probably heard of the business card. I think it's special, and I've noticed if you can add on the back of your business card just a one-sentence research spiel, what you're interested in, and a picture of yourself. There have been times when I've been at conferences and I've given this to people I'm wanting to network with. And sometimes I'll see them walking away and they flip the business card and kind of have like a aha or a little smile. So that's just something to think about if you really wanted to stand out. This was one way to do it. Yeah, so I'll end on that. And I just wanted to say enjoy the journey.